Well, thank you very much for the invitation to this truly wonderful symposium with collections of people with great ideas, great data, and to you, Banks, happy birthday. I'm going to talk today about combinatorial antibody libraries. Combinatorial antibody libraries are a very large, nowadays 10 to the 11th member collection of antibody binding proteins that are arrayed in a format that allows the experimenter exquisite control over the selection. So a subplot, which I'd like you to think about a little bit, if you had access to and control over 10 to the 11th binding proteins that could be used outside the cell or nowadays inside the cell, what could you do in terms of therapy or phenotypic conversion? We can talk about that maybe later even. We call this the chemistry of very large numbers. And the reason we think of it that way is because, and this you'll see much of later, is that we don't want, as people wanted in the past, just a few specific antibodies, but we want hundreds or thousands. And the reason we want so many, and the field wants so many, is we would like to survey a large array of binding energies in order to ascertain how biological consequence is achieved. So in the past, if you had one antibody, you had two antibodies, and you could do a Western, or you could do this, or you could do that. That was good enough. But that's not good enough for the chemistry, the immunochemistry of antibody molecules. So really, then, we're interested in diversity. And diversity is different than large numbers. Large numbers just says that you have in a test tube a large number. But what's really important is that you have a large number of different molecules. A large number alone doesn't mean very much. And so we want to exploit this diversity now using combinatorial antibody libraries, which in essence is a synthetic immune system, to allow one to exploit diversity by studying the chemical potential of a very large number of selectable binding proteins. That's sort of the operative phrase. What is the potential to the experimenter or to the physician of this particular situation? Okay, what's the background? What is the background to all of this? Both immunochemists and chemists, organic chemists, about 20 years ago, maybe a little longer, decided that it would be nice to make a large array of molecules, either very large or small, right? And there are sort of two ways you can think about doing that. One way was that you put some knowledge into it and said this or that scaffold, this or that collection of atoms will make it druggable. Or alternatively, you can make it very large. You're still using some information, but make it very large. That will increase the probability that some molecule in your collection will have sufficient binding energy to be useful for your purpose. I'm sorry, I didn't want to do that yet. So the chemists still today are working on these concepts and trying to measure diversity. And nowadays, they generate libraries of several million like molecules. So if you go to a pharmaceutical company and they want to show you something, they will proudly show you their robots and access to 1 million, 2 million, maybe even 5 million molecules. Nature has solved this call for diversity in defense by a process of gene shuffling and permitting the combination of very large numbers of heavy chains and light chains of immunoglobulins, and package them in such a way that they can select. So in reality, it's a genetic diversification system, A, and B, a packaging exercise, evolution, that allows you to select that. And we tend to have about 100 million antibodies in our bodies. Now, the basis of what we needed to do, the blueprint, if you like, was laid out by the pioneers of immunochemistry. And Professor Porter, Rodney Porter, and a late friend of many of us, Jerry Edelman, told us a lot about the antibody scaffold. And the things that are pertinent to us are the two-chain molecule. It's got an effector function, and the diversity is present at the very tips. At the time these gentlemen were doing this, there was another problem the immunologists called the generation of diversity. Where does the diversity come from? So for instance, if you're interested in trypsin, there are not 10 to the 8th trypsins, there's 1 trypsin. But here, there's a lot of diversity. That problem was essentially solved by Professor Tonegawa, who showed that it's a combinatorial problem. And basically, the diversity is given by a genetic process, which is a VDJ joining a number of heavy chains and a number of light chains in the genome. And because these junctions are irregular, you get a large diversity. Now, one concept which will be important to us later that I'd like you to put in your pocket and recall it when we need to talk about it, and that is because of this junctional diversity, the major diversity and the love object of most immunochemists is the CDR3, one of the loops of the heavy chain molecule, okay? And if you have a long immune response and you look, you will see a lot of somatic mutation, and that's where the game is played. So I put this little red thing there to remind me to tell you that. It's an interesting device. All right, so what are the conceptual and practical issues? So as I said, from the history, we had a blueprint. We knew what genes we needed to generate a proper antibody immunoglobulin. These words are exchangeable, I think. But it's the scaffold that incorporates large diversity. Having said that, there were big problems. If you read the literature before 1989, you will see that many great scientists publishing papers of expression of antibodies in E. coli, but only one chain. And the problem became known as the two-chain problem. And the two-chain problem, sort of like Michael said earlier today, had some truth to it that it was in fact a problem, and some things that were wrong about the concept. And one of the real problems was that the question was, how do you associate these chains in very, very large numbers without getting some sort of scramble, okay? That was a real problem. The not so real problem was the erroneous concept that you need the original pairs. As I'll show you in a moment, in our immune systems, we have one antibody, we have 10 to the 8th, but each one is packaged separately. So you wouldn't buy 10 Christmas gifts and put them in one package and throw them on the floor. I mean, it's a packaging exercise. And so this packaging exercise and the stoichiometry problem, which was a real problem, was an issue. So, for instance, you can take a phage mid, you can put in 10 to the 5th different light chains, 10 to the different 5th heavy chains. That's a combinatorial of 10 to the 10th. send the whole system to the periplasmic space of E. coli, and if you understood the packaging problem, you understood the stoichiometry, you could get them to assemble. Let me remind you that, you all know it, but let me ask you to think about it again. The volume of an E. coli is about 10 to the minus 15th liters. Periplasmic space is much smaller than that. And so if you have a volume of 10 to the minus 15th liters, do the math, a few hundred molecules is micromolar. And the formation constant, studied extensively by immunochemists for a long time, between the heavy chain, those two chains, and light chains, is nanomolar. So if you can figure out how to get the heavy chain and the light chain in the periplasmic space, you can make antibodies. So the requirements to make a synthetic immune system now, assuming you could solve this construction problem, which we did solve, is, first of all, tolerance. This is the central rule of immunology. And that rule states that, it's not a hypothesis, I think it deserves to be a rule, that you cannot make antibodies to yourself. And since almost all therapeutic antibodies, 90% of therapeutic antibodies, are to yourself, because you want to get rid of something, you want to perturb something, you want to knock off a cancer cell, you had to get around that problem. The system has to be clonal, because you don't want a huge mixture anymore. You have to have numbers equal to or exceeding the natural repertoire. There's no sense making a system in immunology, immunochemistry that's smaller than what we have, except for access reasons. And so it has to be bigger. And this is the key. One way or another, recognition and replication have to be linked for access. So the way you do it is you've got these 10 to the 8th antibodies. Each is in a separate package, as I'll show you in a second, that responds to antigen. And so your trick is packaging. And of course, the genes for your antibody are inside the cell. So this is just a depiction of that. We don't have to spend a lot of time. You've got 10 to the 8th cells in your spleen, let's say. It's not quite right, but almost. And each one makes one antibody, like this. And ultimately, you have about 10 to the 8th combinations. So here's the packaging device, again. And here's your spleen. It doesn't really look like that, but each one makes a single antibody. Mind you, one drop of SDS ruins everything, okay, because it destroys the packages. Synthetic immune system is different. It has 10 to the 11th members, different members, and they're contained in about 250 microliters. So what's the problem? Problem is that combinatorial antibody library construction is the opposite of cloning, because you make up some heavy chains and light chains, and some people think they know what the best kind are. Some people just do it randomly. And so you have, nowadays, 10 to the 11th antibody molecules in solution. But you ruin the essence, because this is a test tube. And so now recognition and replication are no longer linked. So what can you say are the principles at this point? Cloning takes complexity, that's what it does, that's what the word means, and reduces it to a singularity that permits selection of an antibody. Initially, the combinatorial libraries were the wrong way around. They converted singularities to complexity. And this is polite company. I won't remark on what people thought of us to take this beautiful spleen and wreck it, okay? So in order for it to work, you must return complexity to singularities because that's the basis of the selection. So now, as I told you earlier, we have 10 to the 11th antibody genes in solution, and they must be repackaged. Now, these are some of the repackaging devices. The original was lambda. Yeast is very, very prominently used. Even the ribosome is used. But the central commonality of all of this is that the genome, the gene, and the phenotype are linked. So if you take M13 phage, for instance, the single-stranded DNA that encodes the antibody molecule is inside, and the antibody molecule is displayed on one of the 5 so-called gene 3 products that are used for the phage to bind to E. coli to infect it. Now, one sub-concept here, which is often misunderstood, which came out of the solution to the stoichiometry problem, is this: that if you, and you could, engineer the antibody molecule into the gene 3 molecule, that won't work because the phage won't be infectious. Because this is the receptor, the ligand that uses the receptor on the pilus in E. coli. So just technically speaking, in one sentence, you had to use a phage bid system in order to be able to control the stoichiometry. And this is what it looks like. This is a selection of an antibody that binds the hepatitis B particle from a patient that was infected with hepatitis B. This is the single-stranded phage we're talking about. It's about a micron long as the phage itself, and it has 2,700 copies of a helical 50 amino acid long protein. The whole thing is helical. And it has on the tip the so-called gene 3 protein, and that's 5 sites, where you could put an antibody molecule if you knew how to make the construction. I show you this not because hepatitis B is so special, it's a bad disease, but I wanted to show you the relationship between this micron-long rod and the hepatitis B particle. And you see each rod binds, for the most part, to one hepatitis B virion, and that's because basically each one of these had one molecule on its tip. You can engineer it in a different way if you like, and you can put the antibodies along the shaft, and then you would see these hepatitis B particles lined up along the shaft. So now you've got 10 to the 11th of these phage particles. Genotype and phenotype are linked, and you want to select them for binding to something, or nowadays for infection of cells to get a phenotype. And this, I think, may be familiar to everybody in one form or another. This is the workflow of how it works. So let's say you have a black, green, red, and green protein. And so what you do is you select for binding, wash either stringently or not stringently. You go around 5 times, and you select for binders. And let's say now this guy was sort of non-specific, it drops out, and now you have 2 antibodies. Actually, you usually have thousands. And then you clone and you isolate now using genetic methods the antibody that encodes the antibody that binds to the red guy. And that's the workflow. In the parlance, it's called padding. It's from the gold miners, I guess. So combinatorial antibody libraries allow creation of a synthetic immune system in a test tube. They use the exact principles of the natural repertoire, where genotype and phenotype are linked in a package. The package happens to be a B cell. They eliminate the century-old tradition of immunization. In this game, there are no live animals used. We can talk about that later, but it's not for animal rights reasons. Most importantly, you circumvent immune tolerance which, as I told you earlier, is important because most therapeutic antibodies are against self-antigens. Since your memory of what has happened to you your entire life, immunologically, is contained in your bone marrow, basically, you contain a history of all the antibodies you made in your life. And lots of times, you'll see later, we want that history. We want to know what you did about Lyme disease, or what did you do about bird flu, or so on and so forth, right? And sometimes these rare antibodies are critical for survival. So let's divide it into chemical considerations and biological before going to a real example. It's the largest diversity system known. I suppose people who are interested in smell could argue this, but probably not very effectively. With the ability to present all kinds of binding surfaces and cavities for antigens that can be very diverse, shape and size and so forth. There are antibodies in there that have pockets, deep pockets, right? Entropic issues. There are antibodies that have grooves for peptides. And then there are antibodies that have 700 square axes of surface binding energy, in order to bind flat to proteins. So those are the categories. As you will see, the use of the large numbers allows one to understand the chemistry of antibody binding energy and synthesize superior molecules. This is very important. Immunochemistry then has moved from an observational to a synthetic science and now allows for comparative immunochemistry. If you've got two hybridomers, you can't do comparative immunochemistry. These molecules that you select usually have very high binding energy, picomolar to nanomolar, sometimes femtomolar. And the reason is, we can talk about it later if you like, they're not constrained by parameters that control things in vivo. Basically, everything in the immune system is yin and yang, everything. It allows you to turn on. You got an equally complicated system that shuts you off. So you have a turn-on system and a turn-off system. Nanomolar seems to be about as good as you could do it in vivo. You can design selection systems, and let's spend a second on this. Move from what has happened, that's what immunology was, immunochemistry, memory, to what can happen, what responses are possible. You're not feeling well, not here, but in the East Coast of the United States. Go to the doctor, if he's a smart guy, a smart lady, they'll say, "You've been crawling around the woods in Connecticut?" You say, "Well, by the way, yes, I did." They will then take some blood over a period of time and see if you have antibodies to the tick. And if you do, and they go up, it's a measure of what has happened, what happened, you got bitten, and now you make the diagnosis. In this game, we want to know what can happen. And specifically, we want to know what can happen if you have 10 to the 11th binding proteins in a selectable format. Method is general. I won't dwell on that. But I will dwell for a few seconds on this. Now, just a footnote to this whole thing. Many experiments now, mostly published, that are quite dramatic in the different way they use these numbers. For example, there are agonist antibodies that will turn stem cells into brown fat, and there are agonist antibodies that will cause tumor cells to start to kill each other. They're very dramatic examples, but they don't speak to the central concepts. They're just a great antibody, right? The only concept they speak to is, other than cell biological consequences, is the fact that the large numbers allowed you to find them, right? But here is a really interesting question. If there were as many immunochemists as there are you in this audience, and you say, well, how does the system work? How does the immune system work? They said, oh, we know all about that. You've got 10 to the 8th antibodies, and if you're infected with Ebola virus, you start an immune response, and you scan sequence space, and you got 23 amino acids in that CDR3 I told you about, right? And you get a good binder, and then he takes care of everything for you, right? But you only have 3 days to live. How can you scan sequence spaces in 3 days with a division time of 24 hours? So there has to be something else. And it's either evolutionary design or luck. Okay, one or the other. Okay. And so what does an emergency immune response look like when there's not enough time for the infected patient or anything to scan the entire sequence space in real-time evolution of the antibody molecule by somatic mutation? Big question. So I was reading, I hasten to say, because this is essentially a chemistry meeting, my wife's New England Journal of Medicine. He's a physician, and there appeared an article by Dr. Lerner describing in 2006 an epidemic of avian influenza, bird flu, which is back, by the way, that occurred in Turkey. 12 children were infected, and if any physicians in the audience, you don't want to see this, and 6 children died. Okay. So we were able, with a lot of difficulty, to get into Turkey and get bone marrow from the 6 children that lived. Why bone marrow? Because that's where the memory is. And it took us a little while politically to get in there with permission from Professor Lerner. From the 6 children, we made libraries. The total diversity was 4 times 10 to the 9th members, right? Greater than 300 of them, clones, were recovered in a single selection that basically means one patient, of which 146 specifically bound to H5HA. Given the dynamics of selection, that means there's probably 10 times more in the bottle. But right now you've got 146 to work with. The affinity antibodies from these young children range from single-digit nanomolar to 13 picomolar. And they had mutations, of course, but the mutations use different codons, right? And therefore, they weren't the same, and it was an example of convergent evolution. So when you do these kinds of things, I guess it's sort of obvious, maybe not worthy of a slide, but there's a selection involved. And the selection depends on what you ask it to do. So if you ask it to bind to peanut butter, that's what you select for. Or good cheese, I guess. But here, we are asking to bind broadly. That is to say, to bind to many members, in this case of group 1, okay? So there were antibodies in these children that did something you rarely see in immunology. You do today now because people looked for it, and that is to bind to a large number of hemaggglutins, some of which go back to 1938. And so if you look at a person, one of these children, you're able, first blush, to look at 115 unique heavy and light chain combinations in one patient. And you saw here that there are some mutations that are required. In other words, if you take those mutations out, you don't have an antibody anymore that binds to bird flu. That's either because they're important or this patient is lucky. Okay. The answer came when, remember, bird flu was a hot issue. and people worried about it being epidemic. And so many people made combinatorial antibody libraries from people. We did, the Crucell Corporation did, and Professor Morisco at Harvard did. And you saw something you never see in immunology, and that is everybody had the same mutations. Now, if I don't have a license anymore, but if I immunized everybody in this room on the way out, chances are no two of you would make the exact same antibody, because your potential is basically 10 to the 8th, right? So you would do that. So then we were fortunate enough to have people like Ian Wilson and others, and they solved the structure of all these antibodies. And there they are. That's the name. There's 4 antibodies, and they're unusual, because I'll show you in a second. The game is played up here before this time. And I'll show you why in a second. And that's the reason that we get a flu shot every year. Because there's a chess game played by influenza and you and me. And that is, the immune system makes a move, the virus makes a move. And they keep moving. And the virus is smart enough to put the targets in the loops here, right? Because it can change those loops. It doesn't matter. Okay. But here, this is a very dangerous place for the virus, because this is the functional reason of the virus. This is functional, if you want to call binding functional. But this is the fusion portion. That's how the virus fuses itself inside cells during the process of infection. All of these antibodies stop the fusion function of the virus. So they are to a functional region of the virus. Did I say, just a second here? Yeah. How do we think about these antibodies? The antibodies neutralize the group 1 virus broadly by binding to the unmutable stem, which is good enough for survival. It's a way of buying time, really. I showed you that, I won't show it to you again. And here's an interesting analysis. This is the flu hemagglutinin based on about greater than 13,000 hemagglutinin sequences. And I think it's probably obvious to everybody in the audience. You can draw a line right here, and you're measuring sequence variability. Almost all of the sequence variability, except for a couple patches here, are in the top half of the molecule. So before the year 2008, the entire focus was on these mutable sites of the virus that vary from season to season. Nobody looked down here, okay? And so the idea was, well, so what? You get a needle stick every year. What's the big deal? And therefore, I know that the virus is going to change all over here, but I just make a new strain next year, right? After 2008, and to this day, the vaccine, and particularly the universal vaccine game, is being played down here. And because these broadly neutralizing antibodies point to these non-mutable sites. So this is the current strategy to make a universal vaccine. Now I said they're good enough, and we can talk about it, and they're quick enough to allow you to live. But then, 2 weeks go by, and now the immune system can work the way immunochemists sort of thought it works, and it does. And later on, you can evolve a rare but much more sophisticated antibody that now even crosses the groups, takes care of most of group 1. These are just these are serological strains, and now has moved into H3 in group 2. And it works in a very different way. It doesn't work here anymore, down at the bottom anymore. But instead, it inserts a long CDR3 in the sialic acid binding pocket, which is now a total steric block of the ability of this guy to make to bind to sialic acid. Now it's unique and it's rare, but it's for next time you're infected. You didn't handle this thing before you evolved this antibody, you wouldn't be around to handle anything else. So now we understand from the immunochemical teachings about the time problem. There is a presence of certain prefix germline frameworks that shorten the real-time evolutionary search of sequence space. Mind you, we didn't search a large number of loops, right? But 2 positions, right? The usual CDR3 mutations, and that's why I spent a little time talking to you about the VDJ joining, is not necessary, solving the time problem. And these mutations are for next time, the so-called immunological memory. And we call these guys the SOS component of the repertoire of the germline. And as it turns out now, studies of others have shown that the SOS component is usually played by one germline. There are 50 germlines. This particular one is VH169, and it is the most hydrophobic germline. The antibodies are universal because they attack function. But the analysis, the way this was done by analyzing a large number, right? And that's because, as I told you earlier, one is analyzing the entire immunochemical history of a patient at the nucleic acid level. It doesn't matter whether the patients are currently producing the antibody protein or not. You're not going to the blood, you're going to the nucleic acid memory. Last slide.
Good.
So this is the combinatorial antibody libraries. They allow the creation of a synthetic immune system. They circumvent the problem of tolerance. The large numbers allows rare events. I didn't talk to you about it, but there are several therapeutic antibodies, including the largest cellular antibody in the world, developed by Winter and his colleagues, called Humira. Very important now to find targets for vaccine design. And I didn't talk about this at all, but the current rage is the very rapid generation of antibody agonists, saying if you had 10 to the 11th binding proteins and you could put 100 million of those inside cells, what could you learn? These are my coworkers. I especially want to mention Ian Wilson, because I didn't show you all the structures, but many of the structures, almost all in the field, in fact, were done by Ian. These are some long-standing colleagues. This is a different project. This is a very different project. Thank you for your attention.
It's time for one question. Pressing question. Slowing down.
Yeah.
Hopefully we will wake up for the panel discussion. It's very nice. You changed, years ago you called them catalytic antibodies.
Some of them are.
Okay, but now it's just a broader term, combinatorial libraries.
Well, combinatorial libraries they grew out of catalytic antibodies because one needed big numbers.
Okay.
I mean, we heard a lot today about acids and bases, right?
Yeah.
If you just took 5 antibodies, what was the probability that you would get an acid and base in the right place?
Yeah, okay.
That was the whole reason in the first place.
Okay. More later. Okay, good. What's that? Okay. He'll give you the microphone so you hear you. She'll give you the microphone so you hear in the back.
Yeah.
It'll come on if you just, I think.
Okay.
There you go.
It's fabulous stuff. I was wondering if you've, and what really matters is you get these things in the end, but I'm wondering, because these antibodies are normally produced as a process of maturation of the immune system, when you just put them together randomly,
Right.
what percentage of them will actually fold well and make a...
That's a really, really important question, and that's what really, I'll use an American phrase, screwed up the people who didn't do this. They had an idea, which they call the natural pairs, okay? And if you were not a natural pair, you couldn't work. Well, the truth of the matter is, because of tolerance, what you really want is the unnatural pairs, okay? However, a specific answer to your question is that in the developing immune system, there's an interesting device worthy of yeast, okay? You make the heavy chain, and then you make what's called a surrogate light chain. It's we can talk about it over a drink. It's a very funny light chain. It's cut in half, okay? And the function of that light chain is to see if the heavy chain is good enough to pair with light chains. And anybody who doesn't play nicely with the surrogate light chain, there's only one of it, okay? So you're taking 10 to the whatever against 1, that clone is thrown out. The only other place a clone is thrown out if the heavy and light chain pair happens to be antibody to yourself. So there's two kinds of editing, and I think this answers your question, Sue. One is folding, okay? And you know that light chain disease causes amyloid, okay? And then the other is to get rid of an antibody to yourself. But as far as we know, that's the basic editing.
Thank you again. Very spectacular set of work.